We're starting rather simple when we talk about phylogenies, and we'll get more and more in depth as we go. For many of you, this will be review. That's great. But for some of you, this might be a reminder, a refresher of things you had forgotten, or it may be something that you have never gone over before. And so this is really critical and foundational to what we're going to be doing, even if it is rather simple. This is an image from the Baum paper, and hopefully you've already read it and taken the quiz. If not, you should pause it, go back, read paper number 1, and take the little quiz. There's a list of 10 questions and then the answers to follow. If you didn't do well on that quiz, I would recommend watching this video, watching 1.2.2, the next video lecture, and then going back and taking the quiz again. You may already know the answer, at least reviewing the questions again, and make sure you're comfortable with all those questions on that little practice quiz. I'm not going to be grading you on that. However, some of those questions or very similar questions are going to appear on this week's quiz over the material. Okay, so phylogenies are diagrams that show the evolutionary relationship of species. Now, there are many misinterpretations of phylogenies. This is one that was illustrated in the paper. It's very simple. Most neophytes, people that are not very experienced, and some even that are more experienced, will look at this and say, oh, look, fishes and frogs are more closely related to one another than frogs are to humans or to mice or to lizards. The paper makes the point that often phylogenies are misread by reading along the tips and ignoring the deeper relationships. We really need to read them in a clade-like manner. Now, a clade is a group of organisms that all descend from a single common ancestor. So we'll look at the definition of these later in more detail. But the mouse and the human form a clade with a common ancestor right there. That's called the node. Okay, and so they are more closely related to one another than either of them are to any of these other species. All living organisms share a common ancestor. So it's incorrect to say that the human is not related to the fish, but you can say it's more closely related to the mouse than it is to the fish. It's incorrect to say humans are not related to trees or bacteria or fungi. Any living organism is related to any other living organism. That's a fundamental tenet of evolution. That's called common descent and was one of Darwin's main points when he came up with his theory of evolution. The other one was the idea of natural selection, which is one of the mechanisms by which evolution occurs. And so we can rearrange diagrams without changing any of the relationships. This diagram is the exact same and means the exact same thing as this one. And again, if you're reading it left to right, you'd say, oh, now the lizard is more closely related to the fish. But realize we didn't change anything. So if we read it in a clade-like manner, then we won't have any misinterpretations. That Baum paper is actually quite good and reviews many of the things and the concepts that you need to be familiar with. So if you're comfortable with it, great. If not, go back over it, review that paper, and take that quiz again to make sure you are. So let's review some of that material and also go a little bit deeper. Now, when we are looking at species, we are looking at large conglomerations of organisms. And I've done this diagram to illustrate that principle. So in this diagram, each dot represents an allele. And so I've clustered them together to represent diploid organisms, like humans, that carry 2 of each allele. And sometimes when an organism reproduces, just because of chance, they pass down, let's say you have 2 offspring. So this allele was passed down twice, and by chance, the allele next to it wasn't passed down at all. Now, in this case, it doesn't matter because these alleles are identical. This is a homozygote individual, right? But if we looked at the mechanisms, this one was passed down twice. And sometimes organisms don't reproduce and don't leave behind any offspring, right? And so this is a more in-depth look at the process of populations reproducing and eventually splitting into new populations. So here we have an ancestral population only made up of 4 individuals. So in reality, it would be many, many, many more for most species, but this simplifies it a little bit. And some alleles get passed down a lot, other alleles get passed down much less frequency, and it's just by chance sometimes alleles don't get passed on at all. Okay? Now, the ultimate source, as we mentioned in the first lecture, of all genetic diversity, in order for us to have differences between species, is a mutation. And here I have denoted a mutation by this red bar, and then all alleles descended from that we will look at as different, and we're going to denote that by this red color. So now we have some genetic diversity in this population. Now this population broke off before that mutation occurred. So they are still exactly like the ancestral population. Okay? And then this allele becomes more and more common. This seems to be a strong signal that there's some natural selection for this allele, that it seems to be really beneficial because it's passed down much more frequently than the black allele. But whatever the mechanism, eventually we get differences in populations because of that mutation. Natural selection, genetic drift can also play a role. Okay, and we'll talk more about those and their ramification on genetic analysis in later discussions. But what this means is eventually, because this mutation becomes more and more common, it comes to define this group of organisms that then further splits off into species B and species C. So if I come along and sample an individual from species A, sample an individual from species C, and one from species C, and I look at this gene, these alleles, I would see right away that species B and C are more similar to one another than species A is because of this mutation that they have both inherited and became spread and fixated in those species. Okay, we'll talk about definitions and what these are and how we use them. This is a simplistic look at how this occurs. There are many things that can complicate this and make similarities between species perhaps not reflect that common ancestry, that common mutation that they both inherited. But we'll save those complexities for a little bit in the future. But I wanted you to see what's going on at the population level. Now, near the end of the semester, later in the semester, we will start looking at population genetics. And essentially, population genetics is looking at what's going on in a single reproducing species, at how this diversity changes, how we can use this diversity to estimate population size or the health of the genetic diversity, at least, of a population. And so that basically, the speciation process is the boundary between population genetics and phylogenetics. And so phylogenetics, really, we're only going to be looking at differences between species, and usually we only sample one species, one individual within a species when we do phylogenetics. Okay, but we will come back and look at this. So I wanted you to start to think about this process and what's going on at the population level. Okay, so there is an ancestral population. That's the ancestral population that leads to both species B and species C, and it is genetically diverse. It has multiple alleles for this one gene. And if we looked at other genes, we might have a very different picture, but we're only looking at this one gene. There is the ancestral population for all of the species, right? And it is genetically homogeneous. Everyone is a homozygote. There's no genetic diversity for this particular gene at all, okay? Now, historically, there were different ways that we would diagram and visualize relationships among organisms. The earliest attempt to do this, at least the earliest recorded attempt that we have that's written down and then was used for many thousands of years, is what we call the Scala Natura. The Scala Natura was first recorded by early Greek philosophers, which you really probably could have called the very first biologists, Aristotle and his intellectual offspring. And he did this in a way where he had very simple organisms on the bottom. Here we've got trees, and then animals, and then birds, and then so on up there. Maybe these are bats, I don't know. Those are fish, and then so on, birds, and whatever order you want, but you do it in a very hierarchical manner with simple things first. And once microscopes were discovered, people started putting microorganisms at the bottom. And of course, we put humans, man, at the top. Now, this was as much a philosophical misconception and a social structure as it was a biological way to organize things. And if you think much about it, especially from a modern perspective, you realize this is not a useful or realistic way to organize individuals. And in some cases, this was even used as a way to reinforce social constraints, where we might put peasants at one level, aristocracy at the next level, And then in this course, we have some religion in here with angels and God at the top. So many of these later, especially Scala Natura, also incorporated social and political and religious ideas. And so although it began as a way to classify things biologically, it was not a good way to classify them biologically, and then later took on lots of other social baggage. Okay, but it was used for thousands and thousands of years. And eventually it came to be replaced by phylogenetic or tree thinking. This diagram up here on the left is actually a diagram from one of Darwin's journals and is one of the earliest recorded phylogenies, where we have a common ancestor, which he denoted by number 1, and then descendant species, groups of organisms, A, B, C, and D, that all descended from that single common ancestor. And he wrote underneath this, I think. It's one of the most famous and important simple diagrams, and led to this, began the beginnings of this revolution in how we think about classifying organisms. But even once we had a tree thinking, sometimes there were still remnants of this Scala Natura, or this hierarchical thing, where we have simple things down here with short branches, and then more complex, and obviously man is at the top. So still there's some elements of hierarchy here of simple to complex, and that is not a good way to think about trees. There is some hierarchical organization in phylogenies. However, they don't imply that some are better or worse than others, or that we can put things in any way. This is often based on complexity, and that's only one way that we could look at things. We could look at diversity, or we could look at complexity in metabolism, which many bacteria have many more complex metabolic and fundamentally different ways to gain energy than all of the eukaryotes. And so this is really from a very biased perspective our own. And so a much better way to think about this is through these diagrams, these phylogenies. So here's the full image from Darwin's journal, I think, with the phylogeny and then some explanation about it. There's a nice quote here, but he was one of the early, not maybe the very first, but one of the earliest and perhaps the most influential in pushing this transition of moving away from a hierarchical, strictly Scala Natura, to a phylogenetic way of thinking about phylogenies. And this is critical. Phylogenies are diagrams that show us the history of species. And so they really do fundamentally provide a roadmap, a foundation for organisms and for understanding organisms. And in the Baum paper, they make the point that just as early geography students need to be taught how to read a map, really we should teach early biology students how to read a phylogeny, because they are the roadmap for all life on Earth. And so hopefully you've already had some experience. We're getting better, I think, but you still have many students who come through their high school education, their undergraduate education, their biology majors, and they have very little or sometimes even no background in phylogenies. So if you fall into that category, you're going to need to maybe spend a little bit more time and go through this lecture a couple times and make sure you're clear about what's going on. If you've already got a lot, if you got it all the way back, it makes my heart happy when I hear that students learn about this, maybe even going all the way back to elementary school. But in high school, introductory biology, we're getting better at it. So hopefully you had some background in this. But it is fundamental. It's one of the foundational theories of all of biology. And I would argue that phylogeny and cell theory are the two pillars, the foundation of all biology. Now, there's a very interesting website. I'll pull it up here for you to look at. You can peruse it a little bit if you want to. This is a website that allows us to look at different groups, and you can click and point. You can say, oh, I'm interested in plant phylogeny. You can look at the major groups and their relationships. This one is very, very conservative. So if you're an expert in one of these groups or know a little bit more about them, you might say, oh yeah, this group of organisms is really better resolved than it is here, but you can look at it and it gives you an idea of some of these. You can go up and down the different groups and even go back and forth and explore if you're interested. So it's kind of an interesting, fun way to look at it, and is readily accessible, but also has some good scientific information about different groups. Okay, so this is a good resource. You might want to refer to it from time to time as we're moving through the semester. Okay, so Get back here to my regular. All right. So a phylogeny sometimes is also called a tree because when we draw them out superficially, they look like trees, like plants with branches that then branch into subdivisions, and we have these places where branches come together like on a tree. We call those nodes. But it's also called an evolutionary tree or a phylogeny. Okay, so just be aware that those words are used interchangeably. And most commonly, you will see this square form with either letters or pictures or names designating species. So when you see a phylogeny, these represent species. And they can be extant species that are still living, or they can even be extinct species, although the data upon which we infer and base our phylogenies, which is the subject of the first unit of our class, is limited if we have extinct species, right? We may only have a very limited fossil record, and so we don't have genetic data for species that are extinct, at least if they've been extinct for a very long time. And we may only have a very small portion of the morphological data. But with the data we have, we can often include them in our phylogenies. Okay? So just be aware when you read a phylogeny, you're looking at species at the end, and they can be drawn pointing to the right, or they can be drawn pointing up, sometimes even left or down, but that's rare. You'll usually see them pointing to the right, but it doesn't change the meaning if you just simply rotate them around and change the orientation. Okay? Often they will be drawn in a triangular form, but the square one works a little bit better and is a little bit more easy to read. For print, you'll see this. These are easier to draw by hand, and sometimes just stylistically people choose this. But this means the exact same thing as this. Okay, we have our species on the end. They have ancestral populations here representing where branches come together. Now, in computer programs, and we really won't get into this, but just so you are aware of it, these are done in a kind of a mathematical or a computer language way. They're drawn like this with the parentheses representing different groups. So D and E are grouped together, represented by that common ancestor. C is the closest relative with another set of parentheses there out. So these can be represented mathematically, and those will be used in the software programs that we're using, but those will be pretty much behind the scenes, and you won't see that. So this is really just more kind of put it in your memory and just realize that, you know, we don't put in images into our computer programs, but sometimes we have the resulting software package that will allow us to visualize them this way. Okay, large trees are even harder to diagram. Sometimes to fit them on, they'll make these little. They'll just kind of stretch them around. This is just simply a regular phylogeny that's kind of been wrapped around a central axis for visualization. And then often you will highlight major groups, but realize that some of these are a little bit deceptive, and you need to be able to read trees to figure it out. So they've highlighted some groups here, like this blue one. That's a valid group. And we'll talk about why it's valid and why others are not. So that's a valid group, but they highlighted this pink one also and put a name on it, but it's not a valid group because, well, it's paraphyletic. We'll look at that definition here in a little bit. Okay, so just be aware that sometimes people can highlight and draw things, and typically if it's in a published paper and it's well reviewed, they'll do that in a way with a description of what they're doing and why they're trying to highlight things. Okay. So over here we have 3 monophyletic groups, OAZF3, OAZF2, and OAZF1. Those are all valid monophyletic groups. But we have others over here that are not. And that might be because it's a historical name and they're just trying to point out that it's not valid, but just be aware of that. And let's look more at how we read and interpret phylogenies. Okay, so again, we're going to go back to our very simple phylogeny here. The internal lines are called branches, sometimes mathematically called an edge. There's a branch of mathematics that actually works on understanding and diagramming and looking at analyzing these types of diagrams and relationships. But that's, again, just something we won't really deal with again, but you may see it in the future in other classes, or if you get more in depth into phylogeny. So we will call these internal lines branches. Where branches come together, that's a node, and whether it is a square drawing like this or a triangular one, it's still just a node. The node represents an ancestral population. At the time, it was a single species, and then broke. If these then diversify, then that branch may now represent the divergence of families or orders or classes or even any other major group. But at the time it was a single species, a population that broke into two descendant species. Okay, so that node represents an ancestral population. And you can even be a little bit more specific and say this is the most recent common ancestor of C, D, and E. That node is the most recent common ancestor of D and E. And then there are deeper ancestors as we go down. And often, not all the time, but on most phylogenies, you will see a root. Oh, I've got a root there at the bottom. Okay, let's come back here. I forgot my terminal branch. So these terminal branches, again, they represent species. Sometimes you'll see this one is a range of different terms for. We really won't use these, but they're here just for your reference. Sometimes it's called a leaf, again, using this analogy of trees. Sometimes it's a taxon, taxa, plural. But this represents a single species. OTU is operational taxonomic unit, which is just a very fancy term for a species or a subdivision of a species. So for our purposes, we'll just call it a terminal branch or a species. Okay, and then also we have these roots, right? So often you'll have this branch. It's not always explicitly drawn, but here we have a root on a large square phylogeny with different clades highlighted in different colors. And so sometimes because there are so many species, we take off the names and just highlight them just for communication purposes, so it's not too busy. But here we have the same exact phylogeny drawn in a triangular manner with a root. And so the root represents the most distant ancestor, but still a common ancestor. The root is the common ancestor of all of the species in our phylogeny. And most phylogenies are a synopsis or a summary. We can't include every single species unless we have a very small project, right? So if I wanted to do all of the Drosophila species, then I can maybe do that. That's actually a pretty big project. There are lots of Drosophila species, and I could include every single described species. But if I wanted to do flies, now we're talking in the hundreds of millions of species, maybe tens of millions, but at least too many to do on a single phylogeny, okay? So for most projects, we pick representatives from all of our major groups. And so this has to do then, how do we pick representatives? That's an entirely different discussion that we'll have later in the semester. But realize that these are our overviews or synopses. We have representatives of major groups, but we have not included every single group. In some phylogenies, you will see the branches drawn with different lengths. So here, all of our branches are basically the same length, depending on when they break off. And so this diagram is called a cladogram. That's a good vocabulary word for you to have. Here, let's put that in here just so you can see it. Okay, so a cladogram is a phylogeny that represents only the relationships. The branch lengths are there just for separation and they don't convey any information. That's a cladogram. It's a form of a phylogeny. This one over here is more properly termed a phylogram. I should put it here also. I'll put that down here. Make this all nice and pretty. Okay, so a phylogram shows relationships just like a cladogram. However, in addition, it shows branch lengths that are an estimate of the amount of evolution. So a really long branch length like this means that group is still most closely related to this group right next to it that share the most recent common ancestor. However, this one species has evolved and changed a lot, at least in the genes that are used to reconstruct this phylogeny. So a phylogram is a cladogram that, in addition, shows amounts of evolution in the branch length. And often there'll be a code down here that is, 0.1 changes for per 100 base pairs or per 1,000 base pairs or something like that. So you can read the description of the phylogeny to say what the branch lengths, what the quantification is for those branch lengths. Okay, so no cladogram, no phylogram, and understand the difference between those. Now, as is fairly well illustrated, we did it at the very beginning, and in the bound paper they do it well, you can twist the branching, whether the branch goes to the right or to the left, you can switch that around, and it does not change the relationships at all. So notice in this phylogeny, A is most closely related to this entire group that includes both B, C, and D. And by switching that around, we didn't change that. A shares a common ancestor with the clade B, C, and D. A shares the common ancestor with B, C, and D. All three of these phylogenies are identical. I could draw them with a square and it wouldn't change. Now, if I changed the C and moved it over so it connected in with the B before it connected in with the branch that leads to D, that would be a difference. But notice in none of these phylogenies have we done that. C connects with D, C with D, C with D. They're all identical. Okay? And so don't be confused by the order of the species left to right on the phylogeny. It's how they are connected that are as important. What clades do they belong to?